Shivaya. I am Vrinda S. Nair, and I will be walking you through my project on Pangenome graph construction and analysis, where I focused on Escherichia coli, a widely studied bacterial species. This work explores a modern way of looking at genetic variation, not through a single genome, but through a graph that represents the full diversity of a species. To start with, let me quickly explain the idea of a pangenome. It's basically the complete set of genes found across all strains of a species. This includes both the core genes shared by every strain and the accessory genes, which are found only in some. In this project, I constructed and analyzed a pangenome graph of E. coli, where node represents the DNA sequences and edges show how they vary across strains. And here I use the tools like Minigraph for graph construction and Bandage GUI for visualization and applied graph traversal algorithms to identify gene families and classify genes and understand how genomes differ from one another. Here is what I will be covering. We will start with an introduction, go over the objective, look at the background and tools, then move into the methods I used, then the current status of the project, and finally a summary with references. This is the introduction part. When we study genomes, we usually look at one reference genome, but for organisms like E. coli, which are genetically diverse, that single reference often misses out on a lot of important variation. That's where pangenome graph come in. Instead of one reference, we use a graph that represents multiple genomes at once. Nodes are sequences and edges show variations or overlaps. In this project, I used 15 different E. coli strains and I built the graph using Minigraph, visualized it with Bandage GUI, and analyzed the graph structure looking at nodes, edges, and the graph's density. Then I applied the algorithms to identify core and accessory genes and even structural variations across strains. Then moving to project objectives. The major objective is to construct the pangenome graph for E. coli, then to visualize it to really see how the genomes connect and vary. After that, I wanted to analyze the components that includes nodes, edges, and their relationships. Then we need to classify genes into core that are common across all and accessory which are unique or partial. Also, I explored structural variations, analyzed the gene families, and looked into what all this tell us about the genomic diversity in E. coli. The broader goal is to help microbial researchers better understand bacterial evolution and function. Then, E. coli and many other bacteria go through horizontal gene transfer, mutations, and adapt to their environments. This makes their genomes highly variable. Using just one linear reference doesn't cut it, it can miss entire genes, especially those that are unique to certain strains. A pangenome includes everything the core genome, which every strain shares, and the accessory genome, which varies across strains. This model helps researchers study both what's common and what's different among genomes. Pangenome graphs allow us to map multiple genomes together. Each node is a DNA segment, and edges show relationships, overlaps, or variations. And this model avoids a reference bias and let us detect structural variance more easily. In this study, as I mentioned earlier, I used the mini graph to build the graph and Bandage GUI to visualize it and apply analysis to identify the key genomic features. Then moving to methodologies. Here, first I downloaded the 15 complete E. coli genome sequences from the NCBI database. All are in FASTA format, and then use the WSL to run the Linux-based tools on my Windows laptop. Next, install the Minigraph, wget and git, then concatenated all genome files and picked one strain as the reference. The rest were used as queries. And in between, I must say I explored several other tools also for visualization parts and for alignment parts, but only I think the most efficient among them was the mini graph. That's why I used it. And next, need to run Minigraph to align all the genomes and generate a PAF file which stores alignment data. Then convert that into a GFA file. This is a format that used to describe genome graphs. The GFA has S lines which represent the sequences, L lines which show overlaps, and P lines which are paths taken by genomes. Finally, use Bandage, a GUI tool, to visualize this GFA file and view the pangenome as a connector graph. Then to analyze the graph, use Python, specifically the NetworkX library. S lines become the nodes and L lines became the edges here. And here I use the BFS, that is breadth-first search traversal algorithm, and connected component analysis in order to figure out which nodes appear across all genomes. Those were marked as core genes. Nodes that were only present in some genomes were identified as accessory genes. To understand how connected the graph is, we will calculate the graph density using the formula 2 into E divided by n into n minus 1, where E is the number of edges and n is the number of nodes. A higher density means more interconnected the sequences are. Also, need to look for structured variants. These are things like insertions, deletions, or rearrangements. Here we need to compare the edges between graphs. Missing edges mean deletions, extra edges mean insertions, and rearranged connections pointed to rearrangements. These are the bash commands executed for the creation of GFA file. Here this command shows the conversion from the .fna format to .paf format, and this shows the conversion from PAF format to GFA format. And this GFA format is what we utilized for the construction of Pangenome graph. This is the Pangenome graph visualization obtained through the Bandage GUI. And from this GFA file and Bandage visualization, I can say like there are 4,362 nodes are there, which represents the segments or genes, and 6,069 edges showing how they relate, and a total graph length of around 12.7 million base pairs are there. This gives us a dense connected view of ecology genomic diversity. This is the Python code that we use for the further analysis in the Pangenome graph. First, we need to import the necessary libraries, which includes the NetworkX, matplotlib, community, and pandas. All these libraries are familiar. The only one is the community library. It is used for the community detection within the graph. I will explain it later. And here the first one is the GFA parser function. It is to parser a GFA file and convert it into a NetworkX graph object. That is, the input will be our Pangenome graph. This is the Pangenome graph format. Yes, this is the Pangenome graph, the input file, and we will obtain a corresponding graph for that GFA file. And here, then we are mentioning about the graph statistics, which includes the nodes, which counts the unique DNA sequence segments, then the edges, connection between the segments, which indicates the genomic variation, and the graph density.
And here we are visualizing the subgraphs. The main purpose of this is to visualize a manageable portion. Here I mentioned like 100 nodes. This is mainly because since the entire graph is too large and it's complex to visualize clearly. So here we are using only the first 100 nodes in order to represent the Pangenome graph. And here we are finding the degree centrality, which measures how many edges and nodes are there. Why we are finding that? Because if the centrality is high, it is likely to be the core genes, and if the centrality is low, then it may be accessory genes. That's why we are finding the centrality. And here community is there, community detection. That is, community represents the gene families, functional models, etc. That's why we are finding the communities as well. And by running this code, we obtained these results that nodes, number of nodes as 4,362, edges 6,069, then graph density, and this is the subgraph representation. And these are the top 10 core nodes by degree centrality. So we find this degree of centrality, and we are printing the top 10 core nodes. Then we detected the 56 communities, that is the gene clusters, and the centrality scores and community assignments are saved here in the CSV file format. This is the centrality scores we obtained, and these are the community assignments. That is, from here we can define the gene families and functional models.
To summarize the current status, I successfully generated the GFA file using Minigraph, used the Bandage for visualization, and passed it using the NetworkX. And I extracted the key graph properties like node and edge counts and performed the density calculation. These steps set the foundation for deeper structural and functional analysis. And we will proceed by finding the uh, core genes as well as the accessory genes. To wrap up, this project successfully built a Pangenome graph for 15 E. coli strains, and this study aimed to construct a comprehensive Pangenome graph for these strains and analyze their structural and genomic properties to understand core and accessory gene distribution. Using a graph approach instead of a single linear genome gave us a much clearer picture of core versus accessory genes and structural diversity. And this method helps avoid reference bias and opens the door to better evolutionary and functional genomic studies. And it's especially useful for bacterial species where genomic variation is common and important. And finally, here are a few of the key research articles I referred to throughout the project from PMC, Nature, and Genome Biology. They really help shape my understanding of this space. Yes, that's it about my project. Thank you.